Okay, then let's continue going closer to, or getting closer to what really interests us as cognitive neuroscientists, namely the Bolt signal. But of course, the Bolt signal doesn't happen in a room on its own, but is embedded into a lot of context. And of course, an important form of context for the Bolt signal is the regulation of cerebral blood flow, which, based on the stuff that we just discussed, we can now have a closer look at. As you figure, I'm always looking for the English words because I'm not used to talking about this that much over the course of the year, hence I struggle sometimes to find the words. Anyway, so cerebral blood flow. In principle, cerebral blood flow in physiology can be described in a similar way in an electrical current can be described. So blood flow here being defined as the volume of moving blood per unit time, somewhat generally. And of course being depending on current on the gradient or the potential gradient, or in this case then the pressure gradient. So how much more pressure for blood to flow through a certain vessel is on the one side compared to it on the other side, and the resistance of the vessel. And the bottom line, so one can quantify all of this, namely in terms of the Hagen-Persay equation framework, which we will not look at. The bottom line is that the vessel resistance depends on the vessel diameter, and that by means of the system you actually get a steady flow in the capillaries. So what is a little bit surprising in terms of the blood flow is that in the big vessels, of course, your heart pumps, and in the big vessels there's this pumping motion of the blood still present, but due to the setup of the vascular system at the level of the capillaries, the flow is actually constant and can be upregulated and downregulated, but it's not a pumping movement. So essentially this slide just tells you that we are not talking really quantitatively about cerebral blood flow, which one could. There are different ways of levels, or different levels of blood flow control and for the brain, and these are the central autoregulation and functional hyperemia. And of course we are more interested in functional hyperemia, but one should not forget that there of course is also the autonomous nervous system that plays a role in maintaining a constant perfusion of the brain. So of course central autoregulation processes also play a role. These are the things that you sometimes that you should consider when you do fMRI experiments, how your participants are set up, and that maybe if you want to do group comparisons, that you don't centrally challenge one of the groups versus the other, then you would have differences in the brain perfusion already from this. Of course, in terms of functional specialization and brain mapping, the increase in blood flow in response to local increases, which we already labeled as functional hyperemia, is more important. There exist essentially more empirical evidence on the regulation of blood flow in the brain. So remember these Sherrington results from the late 1900s. They essentially just put a oncograph on the cortex and measured whether this somehow gets bigger. There is more detailed evidence, for example, from this experiment by Guy from 1988 that looks at the regulation of the arterial diameter. And what this graph shows is that there is some sciatic nerve stimulation again in the rat, and it's looked at the somatosensory cortex, and they had a preparation where they could measure their arterial diameter, which you see is going up in response to this sciatic nerve stimulus, the corresponding blood flow going up, but what is not changed is the blood pressure. So it's not that these functional hyperemia is induced by increasing the pressure on the system, but by lowering the resistance, which then increases the blood flow. So that's one example of experimental evidence in this regard. Of course, this is more kind of research that would be done in clinical biological settings rather than in a psychology department like ours. There's also a different nomenclature about cerebral blood flow control that distinguishes the extrinsic control or the extrinsic neurovascular control that is based on the central autoregulation and modulates vascular tone from the outside, and the notion of intrinsic neurovascular control, which is the control of blood flow from within the neural systems based on cortical neurons, astrocytes, and their metabolites, and which is then closely related to functional hyperemia. Yeah, so in a way, central autoregulation and extrinsic neurovascular control versus intrinsic neurovascular control and functional hyperemia are very similar concepts, respectively. And I just put it down under this, but from the one can then also decouple from the metabolic control, which does not need any active cells, essentially. So that's the difference to these kind of control that directly by means of the byproducts of neural activation modulate vascular control and contribute to functional hyperemia. So these are essentially just terms that are more or less well defined, as you can see, that one comes across when reading about cerebral blood flow control. So people talk about extrinsic neurovascular control, intrinsic neurovascular control, metabolic control. So this shows from the Hüttel, kind of a example picture of this. So so what this shows is that you here have kind of a pile artery, so one of these bigger arteries that's still above the cortex, and that you can have extrinsic neurons, which are part of the peripheral neural system that control the constriction of these vessels. And then if you go into the cortex, you have astrocytes and these neurovascular units. Where do we see that? Here. So here we have an interneuron. Here we have a neurovascular unit. So this idea of a tripartite synapse plus vascular contribution. So basically a close relationship, especially close relationship between neurons, here interneuron, and in this case, this is an here anyway, a neuron, and an astrocyte, and the vasculature, which are all spatially very close together, and hence can allow for neuronal changes, inducing changes in astrocytes, inducing then changes in the pericytes of the capillaries, where also some regulation can happen. So the main thing is here not so much to say the regulation happens at this level, but that the regulation of blood flow can happen at multiple levels. And ideally, one would know, of course, for one's specific and one's favorite brain area exactly how the blood flow is regulated in this brain area. However, this is usually then, I think, not known. How should? So here I'm stressing again the notion of a neurovascular unit, or this concept comprising neurons that make, of course, synaptic connections and essentially are also there. Astrocytes, and which kind of ensheath the whole thing, and pericytes, which are these kind of sparse, smooth muscle cells, and the endothelium of a capillary. And then one way that regulation can happen here is that neural activation leads to an increase in the calcium influx, which then from the astrocytes lead to a release of vasoactive substances, which in turn influence the pericytes here, which you can see here. That's what I meant by sparsely located smooth muscle cells. And being able to change locally the diameter of the capillary. Well, so that's the idea of a neurovascular unit. Of course, these are kind of biological concepts that result from, first of all, looking at the histology and basically seeing that, okay, there are neurons and very close there are capillaries and very close there are also these pericyte cells. So this might have some functional meaning. And then, trying to figure out what kind of mechanisms are potentially able to create these neurovascular coupling processes. And then somebody finds that there's astrocyte calcium influx, and then somebody measures that there's vasoactive substance release, and kind of this how these biological concepts come about, and in the end you end up with a PowerPoint slide that is the biological model, like always in biology. And then you get the Nobel Prize if you're a man and over 60. Was a joke. Anyway. So that's some stuff on neurovascular coupling, and now we want to then zoom in more into the Bolt signal, and looking at the Bolt signal from here first a historical perspective. So these processes that we looked at before, so the anatomy of the cerebral vascular system, of course, are part of anatomy research. Then these physiological aspects like extrinsic, intrinsic regulation are part of physiological research. There's some biochemistry research in terms of vasoactive substances. And all of this basically has accumulates all this knowledge and influences what's happening with the Bolt signal. But there's also kind of the Bolt signal history proper, that is described here in this picture by Hüttel. Fairly early on, we didn't discuss the history of MRI yesterday, but this also started in the 1920s, 30s. At the same time, Linus Pauling and Coriel learned about the magnetic properties of blood, which is of course important, and we will see. More work in terms of nuclear magnetic resonance after the war. Important progress in MR imaging actually in the 70s by Lauterbur and also in Mansfield in Nottingham, contributing a lot to being able to create MR images. And then basically in the 80s putting these things together by looking at the the effects of blood oxygenation on T2 and T2 star contrast, or T2 star contrast mainly, and the uptake of scanners in the clinics, better head coils, and then finally the proposal of bold contrast for fMRI and the first fMRI studies that we have already seen. So what this is, in a way, trying to make clear is that, as stated previously, that bold fMRI and fMRI in general did not come out of thin air or clean air in the beginning of the 1990s, but there was a long research tradition that looked at different aspects of this, like always in research. To now think more about the bold signal, we of course have to talk about hemoglobin and its oxygenated and deoxygenated state. And this is just to remind everyone that hemoglobin is the main molecule that's present in your red blood cells, which are called erythrocytes. And this just shows the development of erythrocytes, which are kind of special cells in the human body because they eject their cell nucleus. So they start when being developed in the bone medulla, so in Knochenmark. When they start to be developed, they fill themselves up essentially with hemoglobin, and then eject everything that they don't need, and then in the end they are ready to transport oxygen. And then they live for 120 days, and then they die. That's what erythrocytes do. So this is zooming into the erythrocytes and showing the hemoglobin molecule, which comprises 4 amino acid chains, 2 alpha chains and 2 beta chains, and in addition what is referred to as heme group that also involve ions. And the hemoglobin molecule, in terms of its biochemistry, is fairly well understood. So if you read a biochemistry book, the chapter on hemoglobin will be especially long because, of course, a lot of research has happened on it, because it's, of course, very important for human physiology. So that's why, there's a lot of research on this, and this also links this notion of that you need iron for your blood, because the hemoglobin actually has these heme groups 4, as you see the other ones back there, which are required to enable the hemoglobin molecule to transport oxygen. So this is now an important GIF that is trying to show the effect of binding oxygen versus not binding oxygen. The oxygen molecule itself is not shown here, but what is shown is these amino acid chains, so the alpha and beta chains, and how the configuration of these chains and the heme groups with the ions changes when the oxygen binds or doesn't bind to the molecule. So you see here on the side the deoxy and the oxy state, and the fundamental thing that you should see from this is that the overall configuration of the hemoglobin molecule changes. And this changes, as you can also see, the relative position of this heme group, and this then changing the magnetic properties of hemoglobin. So in its deoxy state, it's referred to as paramagnetic and induces MR signal loss, so it's magnetically active. In the oxygenated state, it's referred to as diamagnetic, and it doesn't induce MR signal loss. So that's the main thing, and the theory is essentially that it changes its configuration, resulting in these changes in its magnetic behavior. How this really works if you change the configuration, that it changes magnetic properties of something, that's left to future scientists to explain to you or something. Now, what this shows is these early works by Thalburn, that without having a scanner at the psychology department, but working more general in nuclear magnetic resonance imaging and looking at the effects on T2 and T1 relaxation rates. What they found here is that the blood oxygenation, if you put samples into NMR scanner, affects the T2 relaxation time, but not the T1 relaxation time. Yeah? So this is essentially work from the early 80s, long before this was used in cognitive neuroscience, of course, that made in vitro this link between T2 relaxation times and blood oxygenation. The real breakthrough then was based on these rodent experiments by Ogawa in the early 90s, which essentially starts from the observation that if you make a T2 star-weighted image of a rat, you see differences in terms of signal dropout if they breathe different air. So what they did is they perfused, they started out from these observations that in vitro, you see these effects, and the question was, can we also observe something like this in vivo? So they had this rat preparation, and they administered 100% oxygen to the rats, so that the blood is really strongly oxygenated, and they had them breathing normal air. And when making the images, they observed these signal dropouts. So these black spots here, this is essentially the fundamental starting point for this, for combining MR imaging and changes in the oxygenation of hemoglobin. And they essentially validated this, that it's really the oxygenated hemoglobin by also doing in vitro experiments where with the gradient echo sequences, so they compared different sequences, but they used a specific kind of sequence gradient echo where they again saw the signal dropout for the deoxygenated hemoglobin. And this then, these kind of rodent experiments that showed that the level of oxygenation and deoxygenation affects this signal, and that you have more signal if you have deoxygenated hemoglobin be present more than oxygenated hemoglobin. This then motivated to use similar imaging sequences that look at the T2 time constant to try this in humans. And these are these images that I already showed you, these photic stimulation in humans, and looking at the visual cortex in 1992, looking at the motor cortex when people do a motor response, and again Ogawa himself also looking at visual cortex. Yeah? And basically based on these studies, the fMRI field then really took off, because before that was not happening. Another thing that is interesting that I figured out when I read the Hüttel is that these kind of studies, they worked much like in the PET setting, where you basically present a stimulus for a long time. So you see this is like 60 seconds, so a minute. There was a photic stimulation, and it was compared to no stimulation over another 60 seconds, which was very much like PET, where you had to administer this radioactivity, and then it was in the system, needed to be taken up, and this whole process was very slow, and you had to, basically people had to do a task for a minute, and then they had to do a control task. Which is not how we do experiments these days, but the fact that you can also, by very brief stimulus occurrences, induce changes in the Bolt signal was also shown quite early on. It's always a little bit forgotten. I found this quite interesting in the Hüttel, that you could essentially induce Bolt signals not only in the way it was done in PET previously, which of course has nothing to do with Bolt, but it had to do with how people were used to doing experiments with humans and how people thought that it would be required to see differences, but that also kind of event-related brief stimulus conditions also induce changes, and this was also known from the early 90s. Because in the 90s, mainly until the end of the 90s, most people used these kind of block designs, and then only the shift to event-related designs happened. Could have happened earlier. So that's basically some background on the discovery and evolution of the Bolt signal. And now to summarize the whole thing and drive it home, again, the Bolt signal and the hemodynamic response to now tie everything together. So what's the story? The story is that at resting state, there is approximately the same level of oxygenated and deoxygenated hemoglobin present, and this inducing MR signal loss, which is then in the active state, because the deoxygenated hemoglobin is replaced by oxygenated hemoglobin, decreased. So the loss is decreased. So the way you have to think about the MR signal, if there's neural activity, the MR signal goes up, but it goes up not because something drives it, but because its inhibition is decreased. So there is less MR signal loss due to deoxygenated hemoglobin in the active state, and there's more oxygenated hemoglobin present in the active state, hence inducing less signal loss. And that's maybe a little bit, given what we've discussed so far, a little bit counterintuitive, that in the active state you have this increase in oxygenated hemoglobin, and the question is, why does that happen? So it's clear that in a way, if there's neural activity, oxygen is required, but if one would think very basic about it, one would think that, okay, now the neurons eat the oxygen, so the oxygen goes away and the deoxygenated hemoglobin increases, but that's not the case. So the question is, why is there actually more oxygenated hemoglobin, so HB, than deoxygenated hemoglobin during activation? And the basic finding here is that the cerebral blood flow increases. Hence, more oxygenated hemoglobin has the possibility to go there. The cerebral metabolic rate of glucose increases, so glucose is eaten up more, but the cerebral metabolic rate of oxygen is not increasing as much as the cerebral blood flow increases and as the glucose metabolic rate increases. So this is what is referred to as the uncoupling of cerebral blood flow, the cerebral metabolic rate of glucose and the cerebral metabolic rate of oxygen. So essentially, more blood comes in, more glucose is eaten, but it's in reference to this, or in relation to this, not as much oxygen is consumed. And there are different theories about why this happens, and of course it's not really clear and no one really knows. So one story for this uncoupling is this astrocyte neuron lactate shuttle, which essentially is based on the idea that the fact that astrocytes need to work harder if there's neural activity, and astrocytes do not have mitochondria, so they can't do aerobic glycolysis, but only anaerobic glycolysis, that this induces this uncoupling, so that the blood flow helps the glucose to go to the astrocytes, and it brings with it a lot of oxygenated hemoglobin, but the astrocytes, they actually don't need the oxygen, they only need the glucose because they don't have mitochondria, and then do their thing. So that's one idea that is kind of a biochemistry idea. There are alternative explanations of why this uncoupling happens, and they don't have as beautiful pictures as we just saw. So other ideas of why this happens is that overall, and also in the neurons, there's an increase in the anaerobic glycolysis to quickly get ATP, because the idea is that aerobic glycolysis takes a little bit more time because this goes over the mitochondria. Another, in a way, very different idea is that it's not so much actually the metabolic consumption of oxygen or glucose, but is that the oxygen is less well extracted from the vessels because the cerebral blood flow increases. So the idea here is that the blood just flows in a given region, flows faster, and hence the diffusion of gases is not working as good, and that this is then increasing the O2 concentration. One other idea is that what is actually observed there in terms of the increase in oxygenated hemoglobin is that where this happens is actually not where the action actually is, but that is a little bit upstream from this location. So it's actually a superfluous perfusion, so that it's maybe not really in a specific location, has to do with the metabolism at this exact site. The other idea is that if it's actually based on this site, that it makes sense to, in order to avoid hypoxia, so too little oxygen further downstream, that it makes sense to increase the supply of oxygen to a metabolically active region more than is actually needed, because then there's still some oxygen available that can be used further downstream. If that wouldn't be the case, then this metabolic active region would just eat up the oxygen, and everything that is further downstream in terms of the capillary bed would not get any oxygen anymore. So it could also be a safety mechanism. Overshoot only for tissue and supply in arterial vicinity. That's every year. I don't know what this is supposed to mean. Sorry. So the bottom line is that there are multiple explanations of why there is this misbalance between oxygenated and deoxygenated hemoglobin during activation. But the finding is that if there's activation, the level of oxygenated hemoglobin increases as compared to the deoxygenated hemoglobin, and that this leads to less MR signal loss, because the deoxygenated hemoglobin induces MR signal loss, and hence an increase in the both signal, or the MR signal better, because the MR signal, if it's used in this context, is referred to as both signal, but it's always, of course, the MR signal that we're talking about. Now, what happens in terms of the both signal is kind of prototypically, it essentially increases and decreases again. So that's so you will come across notions like the hemodynamic response function fairly soon, basically in 2 weeks or so, when we discuss that in statistical methods. And these kind of mathematical models are based on these observations that if you, for example, look at visual cortex average over a couple of voxels or a number of voxels and look at the response in response to a stimulus, you see a kind of a prototypical behavior. You see sometimes an initial dip. We will talk briefly about the initial dip. You sometimes see a post-peak undershoot, but mainly you see a peak. And this is to a brief stimulation, and the time here is roughly around 4 seconds after stimulus onset, whereas in response to persistent stimulation, what you see a little bit more is kind of a rise and then a plateauing and then a fall again. Where actually I have to say, if you really look at MRI data where you have consistent visual stimulation, this plateau is actually also decreasing a little bit over time. But the idea is that in response to brief stimulation, you get this prototypical bold signal time course, whereas for persistent stimulation, a little bit or quite like in terms of the steady state potentials, these individual time courses or these individual responses fuse, and you get this plateau. Then there are these two aspects of the bold signal that have been the center of debate and scientific discourse. So the initial dip and the undershoot. The question is, where do they come from? One theory is that actually, basically in contradiction to all the stuff that where you have an active site, the oxygenated hemoglobin increases, that actually you have certain voxels that actually early on extract more oxygen, and that before the additional perfusion sets in, early on actually oxygen is consumed, and hence the level of oxygenated hemoglobin decreases, so that neural activity happens. This needs oxygen fairly quickly. It extracts oxygen. The bold signal goes down because deoxygenated hemoglobin is in the majority, and then the vascular process kicks in and delivers more oxygen than is actually needed, leading to this to the typical increase. That's what some people say. Some other people don't believe in that at all. This reminds me to tell you about this special issue in Neuroimage. There was, in 2012, there was a special issue in Neuroimage for 20 years of fMRI, and there are a lot of people who contributed to these early developments, wrote quite nice, brief and easy to read reviews, and there you also find one review talking about the initial dip and how people debate whether that's there or not. But there are many more other interesting articles about fMRI in this Neuroimage 20 years of fMRI issue, if you want to learn more about the historical context of fMRI. The other question is, what about the posthumous undershoot? And this is often, so this posthumous undershoot, meaning that there is, after peaking, the bold signal doesn't really go just back to zero, but goes a little bit lower. The story behind this is as follows. So the idea is that neural activity starts, then the blood flow for some time is stronger than the outflow, so that the venous blood volume goes up, and then the neural activity stops, and the blood flow decrease is actually larger than the blood volume decrease, so that in the venous compartment, this is now important here, in the venous compartment there is, so after the capillaries, there's a bit more deoxyhemoglobin, because we're in the venous compartment, than at baseline. And the idea is that it basically pumps up a balloon, which after the process stops, takes some time to empty itself again. The thing is, with this whole thing about initial dip and posthumous undershoot, some studies observe this, some other studies don't observe this. And this here shows an example in gray, where what is studied is the cerebral blood volume. This in gray shows that the cerebral blood volume in the studies that really believe in the balloon model stays up a little bit longer, but other studies actually show that this is not the case. Here, nevertheless, also observe undershoot. So this initial dip and posthumous undershoot, people debate this. It's not really clear. The question is, of course, that this is important to understand what the Bolt signal is about. In terms of the standard model, for example, the standard model that you use for analyzing Bolt data, which is the double gamma model, that actually doesn't that doesn't really have an initial dip, that only models posthumous undershoot. So it's essentially it's not that the community completely agrees on the initial dip and the posthumous undershoot, but that these things actually create research communities and literatures on their own. So you can basically find a lot of papers that argue in this way for or against initial dip and posthumous undershoot. Yeah. Now to finish this, of course, this is also something that you should always keep in mind when seeing these kind of idealistic representations of Bolt signals in textbooks. So something like this. This is more an idealization, a model, an idea. If you really measure the data, in this case, for example, a 2-second hand squeeze in one participant every 16 to 18 seconds at the resolution that you actually image here. Is it maybe a TR of 1 or 2? I guess more 2. You see that it's not that you don't observe this beautiful initial dip, peak, posthumous undershoot, but that you observe essentially the signal going up and down. Yeah? And that this also differs between different repeats of the same kind of activity. So this is, especially if you analyze data yourself, you shouldn't expect always to see these nice kind of textbook figures in your own data. If you average a lot and so on, they might look more like that, but definitely not on a single trial, like for EEG. So this shows basically this cutout, and you can see that overall, of course, in this case, there is a little bit of a going down here, and there's also a little bit of a posthumous undershoot, but sometimes there's also not. So that you just appreciate that the signal is variable, and that from trial to trial and also from participant to participant, you don't always see the same thing. Good. So the last thing about the Bolt signal that we then should discuss are the neural correlates. But again, I think we should do that after the lunch break, because that's quite important, and I think it makes sense to save a little bit of energy for that, so that you become aware of, although we've now always mentioned neural activity inducing neurovascular changes and resulting in this change of oxygenated and deoxygenated hemoglobin in terms of blood flow, we didn't really say what kind of neural activity we actually mean. And the reason for that is that there are actually not that many studies on this, but a very famous study we will then discuss after the break and start at 12 again. Are there questions right now on this? I think it's already fairly straightforward material and no great surprises except for the fact that there's more oxygen when there's activation, but yeah, it's good to know this, and then sometimes feel a little bit nostalgic when in 4 years you again hear talk about the Bolt signal and feel like, yeah, this is also what I learned back in the days. Although nowadays I only chuck my patterns into a classifier and get 56% of correct prediction. Anyway, good. So then see you at 12 again here.